This is just a manual differential stain. It's Diff Quick Protocol Hema 3 staining process. But all of the differential stains are basically modified Romanowski stains. They include a fixative, which is primarily methanol, a primary stain, which is basically a methylene blue or crystal violet, and then they contain eosin. When we label our slides, we need to label them in pencil because the methanol will wipe off or wash off any pen or marker that we might use. So if you stain more than one at a time, you then wind up with a whole bunch of unlabeled slides which are worthless. All we have to do for this is dip it into the fixative. Then once it's fixed, we let it dry. And I actually have these backwards, so we'll just swap those around. And in our little Martha Stewart lab, we have our automatically dried slide. We're going to dip it into the eosin 3 to 5 times, nice steady movements. We found if we do 5 dips in the eosin and 3 dips in the methylene blue, it gives us the best picture, the best colors under the scope. So you want to dip it in and make sure that it covers the slide nice and smoothly. There's 1, 2, 3. Just kind of let it drip off for just a second. Then you can dip it directly into the methylene blue. Oops, this was supposed to be 5. 4, 5. And then 3 in the blue. 1, 2. And again, make sure you've got a nice even stain across it. If it doesn't stain nice and evenly, dip it a couple more times. Then once you get done there, you simply dip it in the water until it's no longer running blue. You may have to change the water a lot of times as opposed to the other stains, which you won't change very much at all. But when the water gets really dark blue, just go ahead and change it. Then I just basically tap off the excess water and let it dry in a vertical position. And then once it's dry, we can put it under the scope and read it.